Hello students and my dear friends. In this session, we'll discuss some of the important MCQs from Introduction to Microbiology. So this chapter will be helpful for lab technician, staff nurse and pharmacist recruitment examination which is conducted by MHSRB, Telangana state. First question, who is the father of chemotherapy? Option A, Joseph Lister. Option B, Paul Ehrlich. Option C, Alexander Fleming. Option D, Robert Koch. Here the right answer is option B, Paul Ehrlich. Paul Ehrlich, he was the German scientist. He is considered as the father of chemotherapy. Why? Because he discovered the salvarsan. So this is salvarsan. It is called as the magic bullet. So this salvarsan has ability to kill the like a spirochete of the syphilis. So Paul Ehrlich, he is considered as the father of chemotherapy. Next question. Which of the following statements about Louis Pasteur is true? Option A, he discovered spontaneous generation. He disproved the theory of spontaneous generation. Option B, he proposed the germ theory of the disease. Option C, he introduced sterilization techniques. Option D, all of these are true. Here, the right answer is all of these are true. So if you see the first option, theory of spontaneous generation. Theory of spontaneous generation, or it is also called as theory of abiogenesis. So according to the theory of abiogenesis, all the organisms in the nature, such as like the life, it arises from the non-living matter. So according to this theory of spontaneous generation, or according to the theory of abiogenesis, so the life arises from the non-living matter. But it is not correct. So here he disproved the theory of spontaneous generation by his Swan neck experiment or SS neck experiment. So that is correct only. Next, he proposed the germ theory of disease. So what is germ theory of the disease? According to the germ theory of the disease, the infections are caused by the germs, that is microorganisms. So that is also like proposed by Louis Pasteur. And he introduced sterilization techniques. What is meant by sterilization? It is the process of killing of microorganisms from the articles, from the surfaces, from the culture media, including the spores. So sterilization techniques was given by the scientist Louis Pasteur. So all of these are true. Next question. Who proposed side chain theory? Option A, Paul Ehrlich. Option B, Robert Koch. Option C, Mullis. Option D, Pasteur, Louis Pasteur. Here the right answer is option A, Paul Ehrlich. So Paul Ehrlich, he proposed the side chain theory of antibody production. He proposed the side chain theory of antibody production. Next question. Who discovered the antibiotic penicillin? Option A, Mullis. Option B, Fleming. Option C, Leeuwenhoek. Option D, Lister. Here the right answer is option B, that is Fleming, Sir Alexander Fleming. In the year 1928, he accidentally discovered that the substance which is produced by the mold, that is fungi, it inhibits the number of pathogenic organisms. So later, that substance isolated and it is named as the penicillin. So that was the beginning of the antibiotic era. So that was the first antibiotic and it was the most used antibiotic and it saved millions of people during the World War II. Next question. Which of the first human disease proved to be viral origin? Option A, Japanese encephalitis. Option B, yellow fever. Option C, dengue. Option D, smallpox. Here the right answer is option B, yellow fever. So yellow fever, it was the first like a human disease which is proved to be the like a viral origin. It was discovered by the scientist Walter Reed in the year 1902 in Cuba. He established that this disease is transmitted through the bite of the infected mosquitoes. So yellow fever, it is transmitted by the bite of the infected mosquitoes. Next question, name the bacteria which do not satisfy Koch postulates. Option A, Mycobacterium tuberculosis. Option B, Anthrax bacilli. Option C, Mycobacterium leprae. Option D, Cholera bacilli. Here, the right answer is Mycobacterium leprae. So here, what are the Koch postulates? Koch postulates are association, isolation, inoculation, re-isolation. So if any organism follow the Koch postulates, then only it is considered as the pathogenic organism. If it doesn't follow, if any organism doesn't follow the Koch postulates, then it is considered as non-pathogenic organisms. So almost all the bacteria, they follow the Koch postulates except Mycobacterium leprae. So Mycobacterium leprae and Treponema pallidum, Neisseria gonorrhoeae. So these three organisms, they do not follow the Koch postulates, but still they are considered as the pathogenic organisms. Next question. Paul Ehrlich was awarded Nobel Prize for his contribution. Option A, in the field of bacteriology. Option B, in parasitology. Option C, immunology. Option D, virology. Which means like in which of the like branch he got, he was awarded with the Nobel Prize. So if you see, the right answer is option C, immunology. Paul Ehrlich, L.A. Mechnikov. So they are awarded with the Nobel Prize in the 1908. Both of them are awarded with the Nobel Prize in the year 1908 for their research work in the role of phagocytes in the immunity and the first selective theory of antibody production. So if you see L.A. Mechnikov, like he proposed this phagocytosis, which is engulfment of bacteria. And as you know, like Paul Ehrlich, he proposed the side chain theory of antibody production. So for these contributions, he was awarded with the Nobel Prize in the field of immunology, in the science of immunology. Next question, what chemical did Lister used to stop the infections? Option A, nitrous oxide. Option B, ether. Option C, carbolic acid. Option D, none of the above. So here the right answer is option C, carbolic acid. So he used carbolic acid. It is a phenolic derivative. So he used carbolic acid as a spray during the surgery. So this carbolic acid, it kills the microorganisms which are present in the operation theater or which are present in the environment so that the infections are prevented. Next question. Who discovered the importance of agar in culture media? Option A, Ehrlich. Option B, Robert Koch. Option C, Louis Pasteur. Option D, Fanny Hesse. Here, the right answer is option B, Robert Koch. So Robert Koch, he was the first person who prepared solid media by using of agar-agar. So in the initially, there were only liquid culture medias. So with the liquid culture medias, like they have faced a lot of problem because if you use the like liquid culture medias, we can see the diffuse growth. In the liquid culture medias, we bacteria show the like diffuse growth. So it is not suitable for to study the like exact morphology of the bacteria. So for that we need solid media. So here initially they used gelatin for preparation of solid media, but it was not satisfactory. So after that he started using agar-agar. So agar-agar it is extracted from the red algae, that is from Geladium species. So agar-agar just it acts as a solidifying agent. Agar-agar has no nutritive value and it is not influenced by the growth of the bacteria and it doesn't influence the growth of the bacteria. Next question. Which of the following is false according to the Koch postulates? Option A, the causative agent must be present in every case of the disease. Option B, the same disease must be produced when the isolated microbe is inoculated into a suitable laboratory animal. Option C, the pathogen must be isolated from the diseased host. Option D, the causative agent must be present in healthy animals. Here the right answer is option D. The causative agent must be present in healthy animals. So what is meant by Koch postulates? Koch postulates are association, isolation, inoculation, and re-isolation. So as we discussed, if any bacterium follow the Koch postulates, then only it is considered as pathogenic organisms. So if you see the first postulate, that is association. So in the association, the organism must be associated in every case of the disease. So the organism must be present in every case of the disease. And second one, isolation. So it means it should be possible to isolate the organism in the pure culture form. And next, third one, inoculation. So if it is inoculated into the laboratory animal, the laboratory animal also should produce a similar type of lesions or similar type of disease. And fourth one is re-isolation. So re-isolation means it should be possible to re-isolate the same organism from the laboratory animal also. So here the first three, A, B, C are the Koch postulates. And the fourth one, the causative agent must be present in healthy animals. It is not the correct one. It is false. Next question. Which scientist used an arsenic compound to treat the syphilis? Option A, Alexander Fleming. Option B, Edward Jenner. Option C, Paul Ehrlich. Option D, Joseph Lister. Here, the right answer is Paul Ehrlich. So as we discussed, so Paul Ehrlich, he was the first person who discovered salvarsan. So salvarsan, it is also called as the magic bullet. It has ability to destroy the spirochete of the syphilis. It is an arsenic compound. So salvarsan, it was an arsenic compound. Next question. Pasteurization of milk was developed by option A, Jenner, option B, Hook, option C, Paul Ehrlich, option D, Louis Pasteur. Here, the right answer is Louis Pasteur. So Louis Pasteur, he discovered this pasteurization. So what is meant by pasteurization? It is the process of killing of microorganisms which are present in the milk. So here in the pasteurization, there are two methods are there. One is Holder method and second one is flash method. In the Holder method, the milk, it is heated at 63 degrees centigrade for 30 minutes. And in the flash method, the milk, it is heated at 72 degrees centigrade for 20 seconds and rapidly cooling it to 13 degrees centigrade or lower. So by this method, all the non-spore forming bacteria, such as Mycobacteria, Brucella, Salmonella are killed. So except Coxiella burnetii. So Coxiella burnetii cannot be killed by this pasteurization because it can sustain even in some of the higher temperatures also. Next question. Bacteria was first discovered by option A, Anthony van Leeuwenhoek, option B, Paul Ehrlich, option C, Joseph Lister, option D, Louis Pasteur. Here the right answer is option A, Anthony van Leeuwenhoek. So Anthony van Leeuwenhoek, he was the first person who observed the bacteria under his microscope. So he named them as animalcules. So because some of the bacteria and some of the parasites are moving, so that's what he named them as animalcules, which means small animals. So here the right answer is option A, Anthony van Leeuwenhoek. Next question. Bacteria such as anthrax bacilli, tubercle bacilli, and cholera bacilli was discovered by option A, Paul Ehrlich, option B, Lister, option C, Robert Koch, option D, Hansen. So here the right answer is option C, Robert Koch. So Robert Koch, he discovered anthrax bacilli, that is Bacillus anthracis, in the year 1876. And he discovered tubercle bacilli, that is Mycobacterium tuberculosis, in the year 1882. And he discovered cholera, that is Vibrio cholera, in the year 1883. Next question. Who is the father of antiseptic surgery? Option A, Joseph Lister. Option B, Paul Ehrlich. Option C, Alexander Fleming. Option D, Robert Koch. Here the right answer is option A, Joseph Lister. So Joseph Lister is considered as father of antiseptic surgery because he was the first person who used carbolic acid as a spray during the surgery to prevent the infection. So carbolic acid, it is a phenolic compound. Next question, study of fungi is called option A, parasitology, option B, immunology, option C, mycology, option D, virology. Here the right answer is option C, mycology. The study of fungi is called as mycology. So study of parasites is called as parasitology. Study of immunity or study of resistance is called as immunology. And the study of viruses is called as virology. And study of algae is called as phycology. And study of bacteria is called as bacteriology. Next question. Who named the small organisms as little animalcules? Option A, Edward Jenner. Option B, Leeuwenhoek. Option C, Robert Koch. Option D, Paul Ehrlich. Here the right answer is option B, Leeuwenhoek. Leeuwenhoek, Antony van Leeuwenhoek, he was the first person who observed the bacteria under his simple lenses. So he named them as animalcules, which means small animals. So by that time, they don't know about the existence of microorganisms. So that's what they named them as animalcules. He named them as animalcules. Next question. The vaccine which is used to eradicate smallpox originated as option A, live smallpox, option B, a genetic recombinant, option C, live cowpox, option D, none. Here, the right answer is option C, live cowpox. So here if you see Edward Jenner in the year 1796, scientist Edward Jenner in the year 1796, he first introduced smallpox vaccine. So here he observed that like the milkmaids who suffered from the cowpox generally like they are immune to the smallpox. So during the olden days, smallpox was the deadliest disease. So here he observed that the milkmaids who suffered from the cowpox, they are not getting the smallpox. So what he did, so he intentionally took the material from the cowpox. So he took the cowpox material from the cowpox source from the hands of milkmaids. So the milkmaids who are suffering from the a cowpox, so from that lesions he collected the material and he intentionally introduced into the James Phipps's body. So 8 years old James Phipps. So after a few days, James Phipps, he suffered from the local reaction and feeling unwell for several days. And after that, he recovered fully. And 2 months later, again, Edward Jenner inoculated James Phipps with the material that is collected from the human smallpox lesion or human smallpox sore. So he collected the a human smallpox material and he introduced into the Phipps body and Phipps remain in the perfect health and he becomes the first human to be vaccinated against the smallpox. So here that is done by using of live cowpox vaccine. Next question. Who developed the first vaccine of the world? Option A, Jenner. Option B, Ehrlich. Option C, Lister. Option D, Antony van Leeuwenhoek. Here the right answer is Edward Jenner. So as we discussed in the previous slide, so Edward Jenner, he was the first person who introduced smallpox vaccine. That is in the year 1796. So because of this successful and effective vaccine, which is developed by Edward Jenner, because of that vaccine only smallpox has been eradicated. Next question. Who invented the hanging drop method to study the motility of microbes? Option A, Louis Pasteur. Option B, Robert Koch. Option C, Edward Jenner. Option D, Leeuwenhoek. Here the right answer is Robert Koch. So Robert Koch, he first of all invented the hanging drop method. So hanging drop method, it is done to check the motility of the microbes. If you want to check whether the bacteria is motile bacteria or non-motile bacteria, we have to go for this hanging drop preparation. Next question. First vaccine developed by Louis Pasteur was used against option A, polio, option B, smallpox, option C, measles, option D, anthrax. Here, the right answer is anthrax. So he prepared the a vaccine which works which is used against the anthrax. Next question: who discovered bacteriophage or bacteriophage? So who discovered the bacteriophage? What is bacteriophage? The virus which infect the bacteria is called as bacteriophage. Option A, Watt. Option B, Ruska. Option C, Walter Reed. And option D, Goodpasture. So here the right answer is Watt. So Watt in the year 1915, he discovered the bacteriophage. So here Ruska introduced the electron microscope. And if you see Walter Reed, he proved that mosquitoes carry the yellow fever agent. And Goodpasture, he gave the chick embryo cultivation techniques, like virus cultivation in the chick embryos was given by scientist Goodpasture. Next question, who introduced the sterilization techniques? Option A, Robert Hooke. Option B, Ehrlich. Option C, Robert Koch. Option D, Pasteur. Here the right answer is Louis Pasteur. Next question, who is the father of microscope? Option A, Ehrlich. Option B, Leeuwenhoek. Option C, Robert Koch. Option D, Joseph Lister. Here the right answer is Antony van Leeuwenhoek. So Antony van Leeuwenhoek, he was the first person who prepared the microscope. So with his microscope only, he observed the various forms of the microorganisms, such as various forms of the bacteria as well as the protozoa he observed. Next question, who is known as father of bacteriology? Option A, Robert Koch. Option B, Louis Pasteur. Option C, Joseph Lister. Option D, Edward Jenner. Here the right answer is option A, Robert Koch. So here Robert Koch, he discovered the most of the medically important bacteria. So he's considered as father of bacteriology. Next question, staining technique using aniline dye was introduced by option A, Louis Pasteur. Option B, Paul Ehrlich. Option C, Robert Koch. Option D, Edward Jenner. Here the right answer is option C, that is Robert Koch. Robert Koch, he introduced the staining techniques. So Paul Ehrlich also applied the stains to the cells and tissues to study their function. But Robert Koch, he used the staining techniques to identify the microbacteria. Next question. All these are contributions of Robert Koch except first option, introduced solid media for culture of bacteria. Second option, liquid media concept. Third option, introduced methods for isolation of bacteria in pure culture. Option 4, discover the acid-fast nature of tubercle bacilli. So here the options are only option A, only third one is correct. Option B, second and fourth options are correct. And option C, second and third one is correct. And option D, only first one. So here the right answer is option B, that is second and fourth. So here liquid media concept, it was not given by the scientist Robert Koch. Liquid media concept was given by scientist Louis Pasteur. So he used liquid media for culturing the bacteria. And if you see the fourth option, discover the acid-fast nature of tubercle bacilli. So Paul Ehrlich, he discovered the acid-fast nature of tubercle bacilli. So only first option, that is in Robert Koch, he introduced the solid media and he introduced the methods for isolation of bacteria in the pure culture. Next question, Who coined the term vaccine? Option A, Jenner. Option B, Crick. Option C, Pasteur. Option D, Watson. Here the right answer is Jenner. The material isolated from which of the following animal was used for vaccination for the first time? Option A, cat. Option B, goat. Option C, pig. Option D, cow. Here the right answer is option D, cow. So here the material which is used for vaccination that was collected from the cow. So here vaxa means cow. So here the word vaccination came from the word cow. That is, the word vaccination came from the like vaxa. Vaxa means cow. So here the material was first collected from the cow. So here Edward Jenner, like he is considered as father of vaccinology. So who regarded as the father of modern microbiology? Option A, Crick. Option B, Louis Pasteur. Option C, Leeuwenhoek. Option D, Joseph Lister. Here the right answer is option D, Joseph Lister. Sorry, option B, Louis Pasteur. So Louis Pasteur is considered as father of modern microbiology. Thank you.